our Vice President of Programs is going to introduce Dr. Parker to you, but I would like to explain how we're going to run this event. Ann Swanson, our VP of Programs, will be monitoring the Q&A throughout this event. And at the end, she will be sharing those questions with Dr. Parker, who has graciously said she will stay and answer as many questions as possible. So I will turn it now over to Ann.
Good evening, everyone. Thank you for coming. I am very pleased to introduce Dr. Karen Parker. She was born in Boulder, Colorado, and raised in Naperville. She's a graduate of Naperville Central High School. Her mother is actually a friend of mine, which we didn't know until we got talking. She is has the endowed chair of the endowed Truong-Tan Broadcast Professor. Is that correct? She is in the Department of Psychiatry and Behavioral Sciences at Stanford, where she leads the Major Laboratories Steering Committee and directs the Social Neurosciences Research Program. The principal goal of her research is to better understand the biology of social functioning across a range of species and to translate those fundamental insights to drive diagnostic treatment and advances for patients with social impairments. She has graduate and undergraduate degrees from the University of Michigan, and she completed her postdoctoral training at Stanford and then joined the Stanford faculty after that. I will now leave the program to Dr. Parker.
Thank you, Anne, for that warm introduction. Let me just get my slides up here. All right. So it's really a pleasure to virtually be in Naperville with you all tonight. As you can see, it's still light out here in California. The sun's still peeking through. My mom and I often talk about the differences in our just it's only 2 hours, but sometimes it makes a big difference. So what I wanted to do to you today is to tell you a little bit about my journey as a woman in STEM, and then also talk a little bit about my research. Let's see. There we go. Okay. So as Anne mentioned, I was born in Boulder, Colorado, and I was raised in Naperville, Illinois, in a family of 4, along with my brother, Drew, and my mom and dad, Bev and Bill Parker. An early STEM influence was my dad, Bill Parker, who had a Ph.D. in electrical engineering, and for much of his work, for much of his career, worked at Bell Labs, which was then in its halcyon days as one of the world's premier think tanks. And so my dad used to preside over coding lessons when I was probably 7. Here he is in the lower left-hand corner, much younger than I am now, playing around with some computer that he bought probably this was in the late 70s, early 80s. And as a reward for, grudgingly participating in coding lessons, he would allow me and my brother to telnet into the Bell Labs mainframe to play all kinds of games. And I guess one of my earliest ventures into being a scientist was using three-way calling, which was not well known to the general public. And we were abusing it by putting together 2 unsuspecting parties, and then we would listen in on their phone calls. And my dad, just for the record, was not thrilled about these activities, but it was sort of the first time that I started my own scientific experiments. And I really think about, he had this really profound influence in terms of, I think, my development as a scientist and being exposed to emerging technologies early in my childhood. And then I want to give a shout out to 2 of my high school teachers, Dr. Marilyn Holman and Kathy Linder, who were critical. I had a little bit of a party girl streak in high school, and they both kind of were good at keeping me on the straight and narrow and letting me encouraging me to explore, what an academic and a fulfilled intellectual life would look like. So I think I turned out very differently than I might have otherwise had I not had both of them in my life. So if you can see here from this picture, I'm wearing a University of Michigan sweatshirt, and I don't think I'm quite a year old. And my dad is also wearing one. My mother is wearing red, which is a little bit of a dubious choice, given that that might signify Ohio State. But she too was a Michigan grad. And we used to joke that I was genetically programmed to go to the University of Michigan. And so the sort of a choice of college wasn't really in question in a lot of ways. I loved University of Michigan as a student, and I think my parents probably thought I was having a little too much fun at times, so I pledged a sorority. I would attend University of Michigan football games, and I was really excited to be at Michigan when the Fab Five, who were this basketball sensation, they were classmates of mine, and so I had just a blast in college. In terms of, thinking about a career, I agonized a lot over whether I wanted to be a medical doctor or I wanted to do research. And I felt really torn because I felt a true mandate to be interested. I was interested in clinical questions, but a lot of the questions I was interested in exploring, we didn't have answers to yet. And so that combined with influence of my aunt, Dr. Holly Craig, she was a professor at University of Michigan. And, as I grew up, but also while I was in college there. And Holly had a really important influence in me deciding that I too wanted to be a scientist. So I loved Michigan so much, I did not want to leave. And my husband jokes that I was on the 10-year plan there because, there was no way I was going to do my PhD anywhere else. And so in the far left is a picture of me with my PhD advisor, Dr. Teresa Lee. And I did my PhD at Michigan in biological or psychology or behavioral neuroscience, or kind of two different names for the same field. And I was really interested in social behavior. And at the time, there was this emerging field of the neurobiology of social functioning. And there were two molecules that really just captured my attention, called oxytocin and vasopressin. And my dissertation work, along with work of a variety of other scientists, helped cement their roles in attachment bonds and parental care in a variety of different species. I really wanted to stay at Michigan for my postdoc, and I was basically kicked out of the nest. People said, you cannot stay here you can't do your postdoc. You've already done 3 degrees at Michigan. And so it was sort of funny how things happen. So I had a boyfriend who was working in Palo Alto for the summer, and my PhD advisor wanted me to stop doing experiments. So she allowed me to go write my dissertation while I was visiting him. And while I was there, I was like, wow, I love California. And I happened to look up David Lyons and Alan Schatzberg, and I had heard David give a talk. And he said, I'm looking for a postdoc. Why don't you come in an interview? And sometimes the stars just align. And I think my interview that was supposed to be an hour ended up being 7. And he basically offered me the job on the spot and said, come do a fellowship with Alan and me. And what was really exciting about the work that David and Alan were doing was it was fairly rare, and it unfortunately still is. What I loved about training with them was that David was a basic scientist and Alan was a psychiatrist. And so I could do work with them where we would do work in an animal model, and then we could, what we call, translate these fundamental insights to patients with whatever we were modeling. So in this case, I was really interested in anxiety and depression. And I spent a number of years of my career doing research in this area. We did work with squirrel monkeys. These guys are pictured here. And I also did work with people with major depression. And the work that we did then was focusing on the neurobiological and environmental drivers of what creates a stress vulnerable or a stress resilient organism. And then I was really fortunate to be recruited to stay at Stanford as a professor. And right around the time I was setting up my own lab, so the way, things happen in science, once you become independent, and especially if you stay where you trained, you need to start charting a more independent path from your mentors so you can be evaluated as an individual, not sort of as a package deal with them. And so I was really interested in continuing to pursue this translational path where I would do work in animals and also do research in patients. But I really missed the work I had done as a PhD student, studying the roles of oxytocin and vasopressin in social functioning. And around the time I was setting up my own lab, two things happened. One was that there were a lot of parent advocacy organizations all over the country demanding increased funding by the National Institutes of Health, or NIH, which is one of the major is the major funder of biomedical research in our country. And there was a hedge fund billionaire, Jim Simons, who set up the Simons Foundation because he felt that there wasn't enough research being conducted in the autism space. And so I was really fortunate at Stanford when these funding calls came out, and Stanford created an autism working group to be responsive to them. And I was one of the only people that knew anything about the biology of social behavior in the psychiatry department. So I quickly got engaged as somebody who could be a biologist that could work with clinicians to start trying to better understand the biological basis of autism. And and I think my particular take on it was, if oxytocin and vasopressin are involved in what we call species typical social functioning, in every single mammal ever studied, these two molecules are critical for pro-social behavior. And my the hypothesis that I had was, well, what happens if there's irregularities in these oxytocin and vasopressin release or signaling within the brain? Maybe that might be associated with autism, and it might help us begin to think about the biological basis of autism and maybe begin to develop pharmacotherapies that could help improve social functioning in people with autism. So what I'm going to do, I'm going to transition. I'm going to tell you guys, this will be hopefully like a 30,000-foot view of the research that my lab has been doing over the past about 15 years. And then I'm happy to take questions on it. So my first interest was, in a lot of cases, there are disorders or diseases where there are good animal models where you can, begin to study a disorder and then use those insights to begin to better understand patients with those disorders. There weren't any good animal models for autism, and I was really interested in developing one to begin to better understand the biological basis of social impairments. And then what I wanted to do was, once we had a sense of what that biology might be, can we then translate those findings from our animal model to people with autism? And then can we use that knowledge from people with autism to begin to start testing new medications in autism patients? So I'm going to give a little bit of a primer on autism. So autism is a significant public health concern. Since 2000, the prevalence has increased by 417%. Autism is rising in prevalence. That would be a whole nother lecture on its own about why that could be the case. But suffice it to say that the CDC just put out in the last year new prevalence rates indicating that 1 in 36 US children now has an autism diagnosis. Autism is diagnosed behaviorally on the basis of two core symptoms. So one is persistent impairments in social communication and interaction, and the other is the presence of restricted patterns of behavior, repetitive behaviors. Autism is male biased in prevalence, impacting about 4 males for every 1 female. social symptoms are often viewed as the most debilitating of these two core features when in large epidemiological studies. Autism is highly heritable, but the disease biology is poorly understood. And so autism, although you can see here, this is sort of infant about the first 2 years of an infant's life, you can start to begin to see symptoms emerging across development. But clinicians, because we have a behavioral diagnosis, clinicians aren't reliably able to diagnose autism until about 2 years of age. And because there are no robust biomarkers, the diagnosis can be delayed. And because you need a specialist, there's long clinic wait times up to 18 months in some places. And so even though the signs of autism could be present fairly early, the average age of an autism diagnosis in the US is about 4 years, and it's far older in economically disadvantaged areas that lack a variety of these specialists. The treatments, the standard of care currently are behavioral therapies which are variably effective, can often be extremely expensive depending on the state that you live in. There are only 2 medications approved by the FDA that treat core autism symptoms, and those are antipsychotics that target irritability. They have negative side effect profiles, and there is not a single drug yet approved by the FDA to treat core autism symptoms, suggesting that developing biologically based diagnostics as well as drugs that treat core features are 2 urgent unmet needs. One thing I want to point out is that this lack of medication option for autism stems from a drug development crisis more generally, particularly when we think about central nervous system or brain disorders. So if you test a medication in an animal model and it's wildly effective, for brain disease, greater than 90% of these medications that worked in what we call preclinical models fail in human clinical trials. Greater than 50% of these medication failures are attributable to poor animal model selection, which suggested to me fairly early in my autism research that we urgently needed better animal models. But the question is, what is better? And so one of my first tasks that I set for myself was to develop a valid animal model where we would be able to translate what we found in our animal directly to human patients. And at the time that we had been doing this work, a lot of the challenges that kids with autism face are in aspects of their brain that don't overlap with mice or rats, which are the sort of typical animal models for human brain disease, for better or worse. And so I started thinking about, well, what kind of animal model do we really need? So we want the onset to be neurodevelopmental, meaning we want the symptoms to emerge not late in life, but we want them to emerge early in development in our animal model. We want to establish something called face validity, which is simply the outward similarity in appearance between the model's attributes and the patient's symptoms. So in the case of autism, we want to have complex social cognition deficits in a highly social diurnal species that has vision as its primary sensory modality. You can tell right off the bat that using rodent models is probably not a great idea for autism. Most rodents they're nocturnal. Some of them are asocial, and they use olfaction as their primary sensory modality, and they do not have complex social cognition, even the control animals. Then we want to establish something called construct validity. So this is simply that there's a similarity to the underlying cause of the disease in the animal model and in the patients. And so there'd been a long tradition in animal research of doing brain lesions or taking babies away from their moms and rearing them in isolation. And that's not how somebody develops autism, right? And so my thinking was this was absolutely not the right approach. And what we want is to have evolutionarily conserved genes and neural circuits so we can, again, better take the findings from our animal model to put them into to patients. And then what we want is something called predictive validity. So this is that if a medication is tested in the animal model, it's safe and it's effective. And I'm going to give you one example where this was not the case. And so some of you may be aware of the cautionary tale of a drug called thalidomide, which was a morning sickness drug that was administered. It was tested for safety and toxicology in rodents and moved directly into humans. So it was given to pregnant women while and then when the babies were delivered, they had all kinds of limb deformities. When researchers went back and tested this medication in non-human primates, they were able to replicate these limb deformities, suggesting that if they had actually tested the medication in a species more closely related to humans, there wouldn't have been all of these people that would have been so adversely affected. And then we want to make sure that there's good welfare. We want to make sure that our animal model has complex social housing and what we would call species typical enrichment, right? So we want, in my case, I wanted, if there was an animal that was exhibiting social impairments, I wanted them to be naturally produced, so not spontaneously generated. And to me, all of these criteria pointed to the value in developing a primate model. So if you think about going about developing an animal model, this is true of any disorder that you can think of. There's a couple ways to go about doing it. You can do a genetics first approach where you would either screen in a large population for genes that were causative for the disease. You could do something like genome editing. You guys have probably heard of the technology called CRISPR. So you can actually edit genes to cause an animal to have a specific disease, or you could do something like selective breeding. There's also what we would call a behavior first approach. And this is thinking about the phenotype or the behavior that your model organism would exhibit, and then trying to identify naturally occurring or spontaneous animals, animals that would have social interaction impairments and potentially restricted repetitive behaviors. And then you can go about also looking for various biomarkers. And so in our case, I was really interested in asking, is the biological basis of social impairments that we would see in an animal the same as we might see in a patient? And so what we did was we adopted this behavior first approach. And I'll explain what that is on the next slide. And then what we wanted to do was to say, can we find biomarkers, so molecules that signal the, in our case, social impairment, so we could then test for the same thing in people. Before I get to the animal model, so when we started thinking about this naturally occurring social impairments, we looked to, as I think I mentioned earlier in my talk, autism is highly heritable. But unlike some diseases, it's not caused typically by a single gene. There are some incidences of autism being caused by a single gene, but the vast majority of genetic causes of autism are what's called polygenic, meaning lots of different genes factor into creating autism in an individual. And a colleague of mine had done some really interesting research suggesting that autistic traits are continuously distributed across the general population, right? So it's not like a disorder where like a disease where you either have it or you don't. So we're talking about traits that are continuously distributed. And the idea being that there's this genetic risk that underlies this autistic trait burden. And so for me, that suggested that what we would want to do is develop methods to identify and study individual monkeys at this quantitative extreme of social functioning in a large monkey population. And I'll walk you through what this looks like on the next slide. So these are actually real population distributions in A and B. So in A, this is a measure called the social responsiveness scale, which I'll talk a little bit more throughout the talk. And then we have what's called a macaque social responsiveness scale. So this is simply the same scale that we were able to develop in monkeys. And then what we did, and these are real population distributions. So you can see that these traits are continuously distributed. And if we target in people the tail of the distribution, these individuals have autism. If we target the tail of the distribution in rhesus monkeys, they have pronounced social impairments that look a lot like autism. And then what we can do is use that information to potentially look for biological markers in our monkeys and then try to confirm that we see the same markers in people with autism. And so when we started thinking about doing this work, there's a lot of different monkey species, and I was really interested in doing work in rhesus monkeys, partly because they're a standard lab animal, but also because they're one of the closest relatives of humans. They diverged from humans about 25 million years ago. Another reason why I was interested in macaques is if you think back to my criteria for developing a good animal model, they're highly social. They have some complex social cognition abilities. Vision is their primary sensory modality. We already know that they exhibit stable individual differences in social behavior across their life. And we had some hints from the literature that there were monkeys that spontaneously showed social impairments. And so I like to joke that Stanford wouldn't let me have thousands of monkeys on campus, but I was fortunate that only about 100 miles away, there's an NIH-funded National Primate Research Center hosted by the University of California at Davis. There were about 4,000 rhesus monkeys. You can see that they're housed in these large outdoor half-acre field corrals. Having these large cohorts allowed us to identify more monkeys at the extreme. They live in similar ecological circumstances to what they would experience in the wild, meaning mixed multi-male female social groups of all ages, and we can study them across the lifespan. I had two terrific postdoctoral fellows who helped with getting this project off the ground. And we developed 3 different ways to identify these monkeys with naturally occurring social impairments. So the first of them was just this brute force go outside the corrals and do these focal animal assessments to identify monkeys that don't look species typical, that are showing abnormalities in social interactions with other monkeys. And we called these monkeys low social monkeys. We also use this thing that I mentioned called the social responsiveness scale, which is a clinical instrument, and it's used for 2 purposes. One, as I mentioned previously, to look at these naturally occurring autistic traits in the general population. But it can also be used in the clinic as a screening tool for autism. And if a child scores high enough, that score will merit clinical referral for an autism evaluation by a specialist. So we were able to develop this measure for use in macaque monkeys in a that's very similar to the one that's used in people. And then we were able to develop a variety of experimental laboratory-based tests where we could test for things like face recognition, which is known often to be impaired in people with autism. And we could also show videos to these monkeys to see if they could understand social cues, which is something that's often challenging for individuals with autism. But we could, using any of these different ways to identify animals, we could identify this autism-like phenotype, and then we were able to establish that within any given monkey, all of these measures are highly correlated, which is what you would want. You would think that you're measuring some sort of underlying construct. Okay. And then what I want to say is that what we decided was that the thing that was the easiest high throughput was doing these outdoor observations because our team got to know the animals so well. So for the rest of the talk, I'll be talking about these low social monkeys compared to control socially competent, or what we'll call high social monkeys. So over the past number of years, I've worked with a variety of different undergraduates, graduates, vet students, postdocs, Ph.D. students, and to really flesh out what this phenotype looks like in low social monkeys. So first, if we look at low social monkeys, they have a much greater burden of autistic-like traits. If we test them on a variety of tests that are relevant to autism, these low social monkeys show abnormalities in species typical perception and reaction to social stimuli, including face recognition deficits. If we observe them unobtrusively in the home corral, they show impairments in reciprocal social interactions, which is actually a diagnostic criterion for autism. And interestingly, one of the sort of predominant theories for autism is a social motivational deficit. And what was interesting in our monkey studies was that we saw a decreased initiation, but not receipt of prosocial behavior, suggesting that these low social monkeys may also have low social motivation to interact with others. We've done medical record reviews and have shown that these low social monkeys are bullied more frequently. It worsens with autistic-like trait burden, and this is something that we also see in the epidemiological autism literature. We also see, we've been able to identify very early in life that there are these subtle social information processing deficits when monkeys are about 3 to 4 months of age, that predict with 100% accuracy if they go on to develop this low social phenotype. And then we also published a paper last year showing that these social phenotypes and autistic traits were highly heritable, much like we see in macaques' human counterparts. So the next thing I wanted to do was to think about how do we biologically detect these social deficits. And what I wanted to do was to look for molecules that might signal social impairments in a fluid that would allow us to readily translate this to from our monkeys to human patients. And so when I looked in the literature, as I mentioned previously, autism is a brain disorder. But most prior autism work to identify markers of social behavior had been done in blood, not because it was the right fluid, but because it was the easiest fluid. And so when people would publish research on blood biomarkers of autism, they usually yielded poor detection and poor reproducibility. So I became quite interested in cerebral spinal fluid, which is a fluid that bathes the brain and the spinal column because it's more representative of brain biochemistry than blood. And I was also interested that I had a bunch of colleagues that were neurologists, and they use CSF routinely to detect other brain disorders, including things like multiple sclerosis and various forms of dementia. And so I became quite interested in thinking, can we take a page from neurology's playbook as we begin to think about autism? And so what we did in this initial study was we studied low and high social monkeys. And then what we did is we measured key biological pathways that had either been implicated in social functioning, in autism, or in genetic syndromes that were highly related to autism. And of course, given my background in oxytocin and vasopressin biology, I was really curious to see if these two molecules that were repeatedly involved in mammalian social behavior would be implicated here. And so if we did simply a discriminant analysis, which meant we put all the biological measures in a hopper and said, can you just tell me if this monkey's high or low social by measuring all these values? The answer was yes. So we could correctly classify most monkeys by just knowing what we measured in their CSF and blood. but that didn't tell us what was important. And so then we did a follow-up experiment where we looked at all of these measures, and we did for any of the STEM people in the crowd, a logistic regression model. And I'm going to walk you through this because this is going to become important as we go through the talk. So on the x-axis is the molecule of interest, and on the y-axis is the probability of being low social. So any blue dots above the dashed line are correctly classified as low social, and any below the dashed line are correctly classified as being high social. And what you can see is there were 3 molecules that reached statistical significance. So then what we did is we said, well, there really should be group differences here if these are going to be true biomarkers of autism. And when we did some further winnowing statistically, the one molecule that came up over and over again was this CSF levels of arginine vasopressin, or we'll call it vasopressin going forward. And that was really interesting to me, and I'll explain a little bit more on the next slide. So a scientist wants to replicate their data over and over again, right? To make sure it's that it's reproducible. And if you can see here when we look at just vasopressin, the low social monkeys, again, are above the dashed line, are correctly classified in blue, and the high social monkeys are correctly classified in orange below the dashed line. So what you can see is that by just knowing the CSF level of vasopressin alone, we can correctly classify almost every monkey in this study. And then the second panel shows that we do see this group difference, which is what we would expect between high and low social monkeys. We could show grooming is an incredibly important behavior in many primate species. It cements social bonds and it maintains them. And what we were able to show is that vasopressin levels are strongly predictive of time spent in grooming, as one might expect. So the lower your vasopressin levels, the less social grooming you do. And if vasopressin is a biomarker, we'd want it to be consistent. And so if you look at the x-axis, these M's are individual monkeys, and the y-axis is the level of vasopressin in spinal fluid. And we found what one would expect. So if we do repeated measures within a given individual monkey, you can see that the error bars around it are really tight around the initial mean data point. But if you look across monkeys, there's a wide range in vasopressin levels, suggesting that there's within individual consistency, but we see a lot of individual variation, suggesting that this could be tracking something. And this is where I think it starts getting really interesting. So in the early days of the oxytocin and vasopressin research, oxytocin got all of the attention. But what people had forgotten was that vasopressin had been critically implicated in male social behavior. So it was involved in attachment bonds, and it was involved in parental care. And so what was interesting when vasopressin kept coming up over and over and over again across all these studies, it made me begin to wonder, wow, this is really fascinating because autism impacts 4 males for every 1 female, right? So does this tell us something about sex vulnerability, for example? I mentioned that we were able to replicate this in many independent monkey cohorts. And one thing that was quite interesting was that when we looked in blood, there were no differences in blood. So if we had only looked in blood, we would have failed to have found this difference, this pretty pronounced difference in vasopressin levels that we saw in cerebral spinal fluid, suggesting that this was a readout of some brain-specific readout. Also, we didn't see any group differences in oxytocin, which is a structurally related molecule. Each of these are very small molecules. They're each 9 amino acids. They only differ by 2 amino acids, but we never got a hit in oxytocin, really suggesting that there's specificity between these 2 molecules that regulate social behavior in mammals. So, the big question, this is sort of the moment of truth whenever we submit grant proposals, people would sort of say, well, big deal, this is a monkey. What does this have to do with people? And this low sociality is a non-pathological entity. Maybe you're just spinning your wheels. And so I got really interested in trying to get cerebral spinal fluid from people with and without autism. And so I was able, one thing that I love about Stanford is it's a very small school. And so you get to know people all over the medical school. And so we were able, when, spinal fluid isn't taken that frequently, but what we were able to do is to piggyback onto a clinical need to take spinal fluid, and we were able to do this in kids with and without autism to ask our initial question. And so what we did, and again, you can see this this figure in A. It's similar to what we had in the monkeys. We had 7 males and 7 with autism and 7 males without autism that were matched within, I think, a 1-year band on age. And what you can see is that by just knowing the CSF vasopressin level in these 14 individuals, we could classify 13 out of 14 individuals. And then we were able to show a statistically significant group difference between children with and without autism. But this is a pretty small sample. And so I had heard about a colleague at the National Institute of Health that was collecting CSF samples, not for clinical reasons, but as part of a research study. So these are medically healthy children with autism that were getting a lumbar puncture only to go into her study. And so, and these were kids that were being extremely carefully characterized. And so, what we were able to do in the largest pediatric cohort to date was we were able to look at CSF vasopressin levels, and we were able to demonstrate a couple things. One, that we could replicate this effect. Two, that it replicated in females for the first time. So this is the first time we're studying females. And what was interesting is because oxytocin had been more closely linked to female social functioning, I was kind of wondering if we'd see something in different with girls with autism. That wasn't the case. So once again, we saw no evidence that oxytocin was involved, but we did see low CSF vasopressin levels in both boys and girls with autism. Um, and because these kids were so well characterized, we were able to use a gold standard autism research diagnostic assessment to show that the lower CSF vasopressin levels were, the greater the social symptom severity. Interestingly, vasopressin was uncorrelated with the other core feature of autism, which is restricted repetitive behaviors, suggesting that there might be other biological substrates that contribute to that piece of this phenotype, and that creating a biomarker panel, for example, might be a way to develop a new laboratory-based diagnostic test. And then again, we went back and looked in blood, and we had a very large cohort. We saw no differences in blood vasopressin levels in children with autism and typically developing control children. And so this really suggested that this group difference was specific to vasopressin in spinal fluid, and that at least in older children, we couldn't back this out into blood. And then the next study that I did, this was sort of my statistician calls this Project Love Child, because I was so passionate about this project. So what I was really interested in was, kids seem to be developing okay, and then they start veering off on this other trajectory. And so it made me wonder, is this biomarker present really early in life before behavioral symptoms first manifest? And I teamed up with a colleague of mine, John Constantino, who's now at Emory, but had been at Washington University. And he had been banking spinal fluid samples from neonatal infants, so infants that were only a couple months of age, well before the first autism symptoms are known to manifest, and just putting them in his freezer. And what we were able to do is I contacted him and said, John, I've got this really interesting data. Can we go and look in your bank samples? And it was a pretty heroic undertaking on John's part. He had 2,000 paper medical records that our team then went and traced into electronic medical records. And then we traced kids in the St. Louis area to see if they developed typically or if they received an autism diagnosis. And so what we were able to do was essentially say, in this quasi-prospective way, we have your spinal fluid, and if we measure your vasopressin levels, can we predict if you're going to go on to get an autism diagnosis? And the answer is yes. So what we saw was that there were these group differences in kids that went on to get an autism diagnosis many months to years later relative. So if they had really low vasopressin levels, they were at risk for an autism diagnosis relative to those kids who developed typically. We did not see a difference in oxytocin levels in spinal fluid. And one thing that I found to be pretty hopeful, as a parent myself, nobody wants to have their kid undergo a lumbar puncture for a diagnostic test. And so we did some initial work. I was collaborating with a neonatologist here at Stanford, and there were children that were undergoing a workup to see if they had sepsis, and most of them don't. So they're a fairly healthy population, but they need imminent treatment if they happen to have sepsis. And so we were able to collect this fairly healthy cohort of newborn human infants and measure vasopressin levels in both spinal fluid and in blood plasma. And what you can see here is that if we measure this very early in life, we have a nice correlation suggesting that blood vasopressin levels could serve as a surrogate for vasopressin in the brain during the early neonatal period. And then maybe blood vasopressin can be used for as a clinical screening tool. And we're starting to, begin to explore that. So the last study that I want to tell you about is some work that what this all sort of added up to. And so when we took stock of the behavioral and the biological work we had done in our animal model and in children with autism, we had the following. We had low CSF vasopressin was a robust trans primate biomarker of social impairment. That made us think that vasopressin signaling in the brain could be impaired in children with autism. The receptor that vasopressin binds to, so a hormone is like a key and a receptor is like a lock, and you have to engage them together to get behavior or to get a physiological response to something. And what was interesting is you can map where receptors are in the brain, and these vasopressin receptors were densely distributed throughout neuroanatomical regions of the brain known to be critical for social behavior, which suggests that if we gave vasopressin, we could directly target these social brain regions because that's where the vasopressin receptors were. And then we did some work in our non-human primate model showing that we could administer vasopressin. The monkeys could actually self-administer it through nebulization. Vasopressin shows up in cerebral spinal fluid, suggesting it's getting into the brain. And in our primate model, it actually fully rescued social abilities in low social monkeys. So monkeys that weren't capable of face recognition, on vasopressin, this recovered their ability. They were able to respond pro-socially to social cues from other monkeys. And so that got us really interested in asking, if we give vasopressin treatment to children with autism, can we enhance their social abilities? And we wanted to give it intranasally because that's a way to directly target the brain. So I teamed up with a colleague here at Stanford, Antonio Hardin, who's a child psychiatrist, and his primary patient population of interest is children with autism. And so we designed what we call a gold standard clinical trial. So it was blinded, it was randomized, and we had a placebo control. And so we had children that were on vasopressin for 4 weeks, or they were on placebo for 4 weeks. And then we unblinded and then conducted the analyses blind as well. And so what we were interested, of course, when you do a first in class medication, this is the first time this medication is being given to children with autism. We wanted to test the safety and tolerability. And so we looked at things like study dropout rate, we monitored vital signs, We did blood clinical chemistry labs. We measured electrocardiogram, so features of heart function. And then we looked at the DOTS, which is essentially a side effect scale that clinicians fill out. And then we also wanted to ask, well, does vasopressin improve social abilities in children with autism? And if you remember, I've talked about this thing called the social responsiveness scale, and we used this as our primary outcome or readout of our trial because we knew it was related to vasopressin in some of our measures we had done in our monkey model. Um, and then we had secondary outcome measures as sort of an experimentalist first. I wanted to see that if we saw a positive outcome in a parent reported measure, I would also want to see the same positive outcome if a clinician was evaluating it and if the child themselves was involved in a computer-based task of social functioning. And I'm just this is published. I'm also happy to talk a bit more about this work. But what was exciting was that not a single child dropped out of the study, suggesting vasopressin was well tolerated. And then when we looked at all these different adverse events that could occur, there were no differences on whether you were on blinded vasopressin or blinded placebo. And then importantly, we saw no significant changes from when before the child entered the study and then after completion of either study drug or the placebo in any of these safety measures, suggesting that vasopressin was well tolerated and safe. And then what we also found very excitingly was that children treated with vasopressin had about a 10, I think it was about how many points is this? I mean, they had certainly a statistically significant increase in social abilities as measured by the social responsiveness scale. We also saw a significant improvement in blinded clinician impression and evaluation. And also excitingly, when we gave kids laboratory-based tasks, so one of them was, here's a picture of somebody's eyes. What emotion are they feeling? On vasopressin, they statistically improved on the ability to read the mind and the eyes of other people. And if we gave them pictures of people demonstrating an emotion, they fared better they performed better on this when they were on vasopressin versus placebo. So what I want to do is just summarize kind of this 30,000-foot talk, and then we'll talk a little bit about one more thing, and then I'll take questions. So what I want to hopefully have argued is that valid animal models are indispensable for understanding evolutionarily conserved biological basis of behavior, and in this case, including the extremes of the social continuum, which represents autism in human beings. I think these animal models hold promise for testing new medications and for translating directly into patients. I'm hoping that we won't have 90% failures if we have good animal models. One limitation of the work, I always like to talk about limitations. We did not study repetitive behaviors in our animal model. And there's only two symptoms, and it's the second symptom. So we really need to be working that up at some point. And people often ask about future directions for the work. So one thing I'd be really interested in doing is, can we use these infant monkeys that we know are at risk for poor social outcomes? I mentioned that these are infants that we can identify that have subtle social processing deficits. Can we treat them prophylactically with vasopressin, and maybe they'll never develop this social impaired phenotype? And this is obviously a study we could never do in humans, right? So that's something that I would be interested in doing. And then also thinking a little bit about this CSF biomarker work that we've done. So we know that CSF, but not blood vasopressin level, is a robust trans primate marker of sociality. We know that CSF vasopressin level could be useful for detecting autism in children that are already symptomatic and in neonatal infants at risk of developing it potentially. But there's some pretty significant limitations to this work. the control children were sick enough to require a lumbar puncture in the first place, right? We weren't getting perfectly healthy controlled children to come in and get a spinal tap. And another thing that really needs to be addressed is we did not have because the work was so difficult to conduct, we don't have a psychiatric or neurological control group. So what our data could actually in turn suggest is that vasopressin deficiency is not specific to autism. It could actually be a marker of a brain that's developing atypically, right? So what we would need to do in future studies is to have a psychiatric or neurological control group to see how specific this is to autism. Right now, we're in the process of doing gearing up to do a multi-site infant CSF collection consortia, where we could, hopefully prospectively replicate our findings. One thing that we're also doing now is trying to develop a biological panel, first in CSF, and then to try to back the signal out into blood to see if we could develop a diagnostic test for autism so that we can circumvent these, long multi-year waits to get an autism diagnosis. And then finally, at least in our initial pilot trial, vasopressin was well tolerated and improved social abilities in children with autism. And, currently there aren't any other medications that are treating core autism features. And so my hope is that this holds out hope for people that are looking for a medication. Limitations were that this was a very small sample, it requires replication, and we don't know the mechanism of action, and that's something else that we need to be addressed. Right now, we have just closed a large phase 2b follow-up trial, which is the largest single-site autism medication trial to date. So we've enrolled over 100 children in our trial. And that the top-line data, I'm hoping will be available sometime this year. And if we have a positive readout, my hope is to be able to commercialize this medication so that it will be affordable and anybody who could benefit would have access to it. And then finally, what I wanted to do is think a little bit about, the stars really had to align, I think, for a woman to be successful in STEM. And there's so many things that are still stacked against us. And so I wanted to reflect a little bit, personality variables aside, I think there's obviously some factors that are intrinsic to any individual in, predicting their success, but the environment plays an incredibly important role in either helping you succeed or putting impediments in your path. So one thing I, wanted to, as I was reflecting back, I had an engineering father with a PhD who never once discouraged me from pursuing a PhD or pursuing STEM. I never heard girls don't do that. In fact, I think the only way I failed him was not being an engineer myself. I had a mom who was a role model. She earned an MBA, MSW, and PhD. I had an academic aunt who was a thought leader in her field, and she made me think that I could be too. I had a wildly supportive PhD advisor who was a wife and a mother, in addition to being a successful and highly respected scientist. And I had two co-postdoctoral advisors, both men, who taught me how to navigate the unwritten rules of academia. They groomed me for success and then brought me into their influential networks that I still benefit from today. And I had a variety of department chairs and deans who provided key opportunities and who supported me every step of my career. And I have a close-knit cohort of fellow female scientists and physicians that I call my friends, and they have been a real source of comfort and, support and cheerleading. We cheer each other on, and we have a very lively text thread that makes me feel supported on a weekly basis. And then I also have a supportive and loving husband who's a devoted father to our 3 boys. And then what I wanted to do, it takes a village to do all this work. So I just listed out some of my early and key influences, and then collaborators, mentees, research assistants, and then technicians, participants and families, and then all of the funding that makes this is needed to make this work possible. And then I wanted to thank everybody for their attention. I know it's getting late, your time, and I really appreciate you guys attending the talk. So I'm done. I am going to stop sharing, and then I'm happy to take questions.
Thank you, Karen. Right now, there's only one question. How
Okay.
long is the treatment improvement after stopping the nasal treatment?
That's a really good question. We don't know the answer to that yet. So I really wouldn't want to speculate, but this is the kind of stuff that really needs to be worked out in detail in the subsequent work that I'm hoping we'll be able to do.
Okay, there may have been another question, but I've been having great technical difficulties. My
Okay.
system crashed like 5 times during your talk.
Oh, no.
So, I don't know.
Question is, did anybody else?
Yeah. I don't know, Lee or Elaine, if you saw an earlier question, because I did and I didn't capture it.
I see one actually here, and it's a really good one. Is there a difference between ADHD or ADD children in your study? What was really interesting, so we don't have a good answer to that. We excluded kids in, I think if this is for the the medication trial, we excluded kids that had really significant mental illness like schizophrenia or bipolar disorder, but kids with ADHD were allowed to be in the study because that's highly comorbid. In this follow-up trial, we might actually be able to look at that. One thing that was really interesting in our infant CSF study was that the infants that also had ADHD did not look the same in their vasopressin as the kids that ended up having a more classic autism presentation. So the kids that had ADHD and other diagnoses, vasopressin wasn't not as predictive as it was in the infants that ended up having sort of more classic autism. So I think that one thing I'll say about autism is that there's a saying that if you've seen one child with autism, you've seen one child with autism, right? It's what we call a highly clinically heterogeneous patient population. And so there's going to need to be a lot of work to make sure that these diagnostics are personalized and that these medications that we develop, like I don't think vasopressin is going to be a smoking gun for everybody, right? It probably will benefit a subset of children, and there are going to be other medications that other kids will benefit from because the biological basis of their autism may be different.
Okay, thank you. Somebody has asked, she has 2 autistic nephews, and she wonders when they can have access to vasopressin.
Yeah, I mean, it's such a good question. Oh man, I mean, we were we were conducting this trial during COVID, and we were down for about 2 years. And the federal government bailed out every single industry except for federally funded researchers. And so what we've been doing is going around trying to get donor money to finish the trial, and it's through sheer force of will of our team that we've been able to you know recruit as many people as we have under incredibly difficult circumstances. So I think, it's not commercially available now. And if you are able to try to get it off label, it's incredibly cost prohibitive if even a doctor was willing to try to write something. So my goal is to get this trial done. And if it works well, then we will try to commercialize it. And then there'll be phase 3 trials that people would be able to participate in, likely in the context of a commercial entity or company.
Okay. Another question. Did you rate the level of severity of your test subjects, and did they respond differently based on that level?
In terms of the medication, the first trial, we had 17 kids on vasopressin and 13 on placebo. And so that wasn't readily possible in that trial. We were just looking at, was there a response or not? In this follow-up trial, that's actually one of our aims would be to see if baseline sort of symptom severity is a predictor of response or not.
Okay. What was your question?
Did I miss perhaps why some children don't have vasopressin? And
Um, this is from my husband, and
Yeah.
since our computer cut out, he wonders if he missed why some children don't have the vasopressin
start,
to start with, or is the not having the vasopressin the reason they have autism?
It's a really good question. So, as I mentioned, autism is highly heritable. And if vasopressin were causative for the disorder, one would expect that vasopressin, the gene vasopressin, would be repeatedly identified as what we call an autism susceptibility gene, and that is not the case. And so what I think is going on, it's funny, I took a slide out that explained it, but I thought it was getting too technical, so that's why it ended up on the cutting room floor. But what I think is happening is people have often hypothesize that, as I mentioned, there's multiple autism susceptibility genes that sum together in any given person, which is why I think the different, phenotypes for any person with autism look really different, right? Because the genetic basis of their autism varies a lot. And what I think happens, and people have been hypothesizing for years, is that regardless of what genetics are involved, these genes converge onto a few common neurotransmitters in the brain. And I think vasopressin is likely one of them. So it's not caused genetically, but that vasopressin is one of the systems that's vulnerable to being impacted. And so you see this decreased vasopressin signaling, and then that may be, contributing to the development of autism.
Okay. Tina Hinch says, excellent talk. Thank you. I just wanted to mention that I worked at the Naperville location of Valve Labs for 30 plus years and recently retired. I also worked on three-way calling, and she believes that she crossed paths with your dad.
Oh, I love that. Thank you for that.
Another question. Do you think that vasopressin would help with adults with autism?
Yeah. My hope is that we'll be able to try this. I was on Huberman Lab, which some of you guys may know about. It's often like the #1 podcast in the world, and it's run by a colleague of mine here at Stanford. And after I appeared in December, I have been inundated with people wanting vasopressin, wanting it for their loved ones. And a lot of adults have reached out to me and said, so much of the research is done in children, partly because I think parents are advocating and it's a new diagnosis and it's when the brain is being, formed. So we think that we might be able to intervene better. But I think it's entirely possible that this could also benefit adults. And I really hope that we're able to do some studies in adults to see if they too can benefit. But so far, we've only done our studies in kids ages 6 to 17.
Okay, and kind of a follow-up question. Is there a better window of opportunity by age for the treatment?
Yeah, we don't know that yet. I think that if we were able to intervene when a kid was 2, I think one of the issues with any kind of medication is, are we looking at a replacement strategy, right? So if a kid isn't making enough vasopressin, then you might think about chronic replacement. Some people have argued with these neuropeptides like oxytocin or vasopressin that they're really more involved in social attention. And so that what we really should be doing is pre-treating before somebody would go into behavioral therapy, right? So that they're looking in the right place for the social cues. And so I think that that's something that we really need to explore, and that may differ by individual, right? It may be that there are people where a vasopressin replacement strategy could be most beneficial. But we're still we're still in the early days. My biggest concern is replicating, this finding to make sure there's a there, at least with the medication.
And another question, is vasopressin given for any other syndromes or diseases?
Yeah, it's a great question. It's given for central diabetes insipidus, which is a form of diabetes when there aren't enough vasopressin neurons in the brain. And then it's also given for an analog of vasopressin called desmopressin is given for um, nighttime bedwetting, for example. And what's been really interesting is that these vasopressin has effects in the brain, but it also has effects in the body. But the medical specialists who study the brain and the body often don't coordinate clinical care. And so one thing that we started, I have a medical student in my lab, and we started thinking about, well, what if people with autism who have vasopressin insufficiency also have medical problems that are related to vasopressin in their body, right? So, people who drink are always thirsty, that they drink, that they do a lot of, they have polyuria, or they wet their bed at night, or they drink lots of fluid. And what's been fascinating is I mentioned this on Huberman Lab, and I've been inundated by people from all over the world who said, I never knew why these symptoms might be related, and now this makes sense, right? And so we've actually just launched a study to try to evaluate how common these physiological symptoms are in people with autism, because I think that they might be an easier way than, say, doing a spinal tap to figure out who might benefit most from vasopressin treatment.
Interesting. And this is from your mom. Is there some way that ordinary people can advocate for you to obtain grants to support your research?
Oh, Mom, I love you. I mean, one thing I will say, my mom hears me complain about this constantly. I mean, at places like Stanford and Yale and Harvard, my husband says that I have a startup with no upside, meaning I have no equity, but like I have to raise all my salary. I have to raise the salaries of everyone on my team, and I have to pay for every penny of anything I do in my lab. And so what's really frustrating is you write a grant, you have to resubmit it. It maybe gets paid 2 years later, and 25% of the budget gets cut. So you have to do 100% of the science and 75% of the budget. And so it's a pretty broken industry. And I say this a lot, which is we would have treatments for a lot of diseases that impact our loved ones if our funding model weren't so broken. That's why I've been so grateful to the donors that have stepped forward and just said, enough's enough. Like, people, if you guys have diseases and you have capacity, like give the money to the scientists who are actually at the front lines doing the work, because like they don't need to be distracted writing grants if they actually think that they can move the needle on a disease that impacts people we love.
Okay, I think that's all the questions.
Right.
So thank you so much. This is very fascinating.
It was my pleasure. It was, again, very nice to be there virtually in my hometown.
We're glad to have you.
All right. People are free to contact me too if they want to. I mean, you guys, if anyone wants my email's very easy to find. It's also on my lab website.
Okay. All right. Thank you.
Thank you. Okay. Good night, everyone.
Good night.